Başla. Recording başlıyor. Webcast. YouTube'da bakıyor. Böyle hibrit biraz karışık oluyor ama deniyoruz. Bir kayıt oluyor. Evet, thank you for coming. This is a Biomedical Engineering Institute seminar organized jointly with Center for Life Sciences and Technology, from Istanbul Health Industry Cluster. So today we have a great pleasure to have here Dr. Aslan, Mahmut Kamil Aslan. He is originally got his Ph.D. Undergrad degree from Relocation University and also master's degree later at the Middle Eastern Technical University, both in electrical engineering. Then went for a Ph.D. at ETH Zurich, working there with the deMello. deMello. This is okay. As far as I know, very well known in this field. He just finished his PhD last summer. He has been still working, keeping his appointments there. Has gonna start his hopefully a postdoctoral appointment soon to be announced. Let's say he's also involved in startup ecosystems. What he has been also doing in his thesis. This is an interesting field which I remotely observed, but personally know very significant. It is intersection of microfluidics, in vitro diagnostics, biosensors, biomaterials. So looking forward to your speech. Thank you for coming.
Yes, I think it's ready, right? Okay, good afternoon. Thank you for the kind introduction. And it is a pleasure for me to be here today. And today my presentation title is Microfluidic Platforms for Single-Cell Manipulation and Analysis, which is basically dependent on the work I have been doing in last 4 or 5 years. So maybe most of you already know about microfluidics, but I just want to give a brief introduction about microfluidics. So microfluidics basically means manipulating or controlling fluids, usually the range of microliters. So it is achieved by designing geometrically constrained channels, which is basically in the scale of micrometers. And what is the advantage of microfluidics? It is cost effective because you are basically using less reagents. For example, if you are doing some kind of some kind of antibody experiment, for example, or some kind of antibody assays, basically it will decrease your reagents and it will dramatically decrease the cost. And it also has high precision because especially if you are doing chemical reaction, you can really do it in a very small volume and it will increase the reaction speed and other precision parameters. And it will also decrease the time for the experiment because you can easily parallelize microfluidic channels. And recently, what is interesting about microfluidics is you can even couple microfluidic chips with some electronic circuits or some sensors. So we call this concept as a lab-on-a-chip devices. So basically, you can perform your reactions here, and you can read or analyze the outputs of the reactions by coupling with an electronic circuit or by putting a biosensor, sometimes electrodes, at the nearby your channel. And it has very wide applications, especially in the last 2 decades. For example, disease diagnostics, drug discovery, or some lab-on-a-chip devices for single-cell sorting, single cell analysis, I will talk about it, or detection of some smaller particles like proteins or RNA, DNA detection. But basically today, I just want to focus on two applications of microfluidics, which are single cell manipulation and analysis. So left part of this cartoon, you can see some kind of single cell isolation and manipulation methods. We can broadly group by passive and active methods. For example, droplet-based microfluidics and hydrodynamic trapping is passive methods. Here, for example, you can have some continuous flow and dispersed flow. So you can basically generate some droplets. This is very well-known, one of the well-known technology. You can encapsulate single cells, for example, and you can trigger some reactions inside of it, some enzymatic reactions, or if you want to track some proteins, you can easily do it with droplet-based microfluidics. And even more interesting, some commercial products are already using this technology. For example, in cosmetics, they are already using this droplet-based microfluidics because with the help of parallelization, you can really generate millions of droplets per second very easily and very robustly. And the other methods are generally active methods. For example, Viscoelastic method. Here you can just apply some acoustic waves to manipulate cells or particles. And here I will talk about in a minute, Bioelectrophoresis technique, for example. This technique is based on some electrical properties of the cells. According to electrical properties of the cells or particles, you can repel or pull the cells, or you can sort them, or you can trap them. And another method is, as you can see, optical trap. Here you can just basically use some laser beam, and you can just exert some force on the cells to manipulate or to sort the cells. And the magnetic method is another method also. But here you need to tag the particles that you want to manipulate. And the right side of the cartoon, you can see some common analysis, single cell analysis methods. Most common one is laser-induced fluorescence, which I will talk about mainly in this method in my presentation. And the other one, we can say the electrical methods by measuring some electrical current or some impedance measurements, we can say. And the other one is mass spectroscopy. You can analyze the particles by considering the mass and charge ratio. So after this brief introduction, I want to talk about 5 different projects. And I can say the first part of the projects are manipulation of the single cells, and the other part is analysis of the single cells. It is more or less like a cytometry approaches. I will introduce 3 different cytometer sorting devices. And first, I want to start with landscape imaging of depth manipulated cancer cells. Depth refers to Bioelectrophoresis, by the way. So Bioelectrophoresis is basically label-free method, and it depends on unique dielectric properties of the cells or particles, and it has simple implementation, and it is also cost-effective, and you can both apply positive on or negative depth force on a particle, dielectric particle. So basic theory behind the Bioelectrophoresis phenomena is we can basically model the cells, including resistors or some capacitors. And we can also calculate the values of both these resistors and capacitors, or membrane capacitor, for example. And after that, because these are the unique parameters for each population of the cells, this factor, this is a more complex formula, but let me talk about briefly. So it depends on the dielectric properties of the cells and the surrounding medium. So it has some complex part also. Therefore, by frequency, you can have different magnitude of the depth force. So it also depends on the conductivity of the surrounding media. For example, this is the graph for the MCF7 cells, which are basically cancer cell lines derived from these immortalized cell lines. So as you can see, until this frequency, for example, if you look at the dark blue graph, until this frequency, we can observe negative electrophoretic force until 12 kilohertz frequency. This means if we use a wave below this frequency, the electrons tend to push the cell. But however, we call it as a crossover frequency. After that, we can, this frequency, we can repel the cells from the electrons. After this frequency, we can pull the cell or particle towards the electrodes. For example, if we consider this graph, at 1 megahertz, you can see we have the maximum deck force. So, for example, if you want to sort MCF7 cells or trap MCF7 cells, it makes sense to use the 1 megahertz frequency of signal. So another technique we use in this project is lens-free imaging. Lens-free imaging is basically imaging, but without lenses. You can think about it like a fingerprint of a cell, because in this imaging technique, we have illumination source, and we are just basically obtaining the shadows of the cells. And the contrast of the image, we can say also quality of the image, is highly dependent on the distance between the object and the plane and the illumination source. And by optimizing it, for example, you can obtain different contrast of image. But if you you can also obtain different size of the image. There is a trade-off between contrast and the size of the image. So in recent years, this approach is becoming very popular because it is low cost, especially when I talk about lab-on-a-chip systems. In lab-on-a-chip systems, in recent years, smartphones have been emerging for the cell imaging. For example, in here, you can see blood image gathered by a smartphone. So as you can see, by using lens-free image, you can really attach very lightweight attachment to a smartphone, and you can obtain decent images of the cells. And what is the advantage of smartphone? Nowadays, you can gather smartphone everywhere of the world. So especially in resource-limited regions, for example, in Africa, if you want to make, for example, malaria detection or other diseases, you can easily use a smartphone instead of using a laboratory equipment. And you can just take the image, and by using a cloud server, you can process the data and you can get some analytic results. So because of that, lens-free imaging is one of the emerging techniques in cell analysis. So in our project, when we consider the advantages of the Bioelectrophoresis and lens-free imaging, we just couple both systems. You can see here we just use a lens-free imaging setup, and here we just use a microfluidic device. This is basically the Bioelectrophoresis device, and it has interdigitated electrode structure. And the whole setup is operated by a smartphone. So this setup, first, we are tracking the cells by using dielectrophoretic force, and by implementing a custom designed PCB, all of the setup can be operated by a smartphone. So after tracking the cells, we can just image them and quantify them. So in our application, you can see from the video, when we apply dielectrophoretic force, we can just track the cells, cancer cells. And we also designed different sizes of the electrode and different spacing between the electrodes in order to optimize the dielectrophoretic force. And we can achieve more than 80%, around 80% trapping efficiency when we are using 2 microliter per minute flow rate.
So which ones are cancer cells? Which ones are normal cells?
Actually, these are just the cancer cell line. This is like a proof of concept study. So when we apply the frequency, they are trapping. When we cut off the frequency, they are just flowing. So second project I want to talk about is image-guided microfluidic system for single-cell lineage tracking. So motivation is maybe one of the most fundamental questions in biology is how cells behave. So basically you can think cell as a structure. It has biosensors around it. It can sense the environment and process the data with their internal network and reach to a decision. However, this process is not straightforward at all because cells have complex internal molecular structure, so that it is not trivial to decide about cells state or examine about the cell's decision mechanism. So in this project, we just aim to track single hematopoietic stem cells in order to understand the stem cell differentiation or self-renewal processes and the decision-making mechanisms during the differentiation. We just want to understand about at the single cell level. Since conventional approach, maybe most of you already know about this. Conventionally, we are just growing cells inside of the flasks. So it is a very straightforward protocol, and it is very robust. You can really grow the cells at huge amounts. But what is the disadvantage of this conventional approach? You rely on population aggregate measurements. You are just basically ignoring the cell-to-cell variability. However, in this, cells are very heterogeneous. So you need to analyze every cell at single cell level. Another disadvantage is coupling this system with some imaging modalities can be tough because you need to do some analytical modalities in order to analyze the cells and some internal molecules. However, recently microfluidic approaches are emerging. So as you can see, for example, this is the conventional cell culture well, and this is the microfluidic culture chamber. You can see one of the examples of the device. So in microfluidics, you will have very small volumes, which allows dynamic control of the medium, environment medium. So you can do single cell manipulation and analysis, and the surrounding medium ratio is much more smaller than the conventional well. Therefore, you will have reduced volume and you can track more precisely cell-to-cell interaction and cell-to-medium interaction. So for this purpose, our goal was designing a microfluidic device for tracking the stem cells at single cell level. So the device should capture single cells, grow them, and separate them. Separate the sister cells, I mean. So in that way, we wanted to understand the role of the gene expression between the sister cells. And also we aim to track the several generations inheritance of a single cell. So for that purpose, we designed multi-layered microfluidic device. You can see the light green color is the control layer and the other light blue is the fluidic layer. And one of the key mechanisms in our device is quake valves. So this is basically like a pneumatic valves. You will have some kind of flexible material. In our case, it was PDMS that can deform when you apply some pressure. So you can see from this video, if we apply pressure, you can control the fluid flow. You can basically open and close the valves. And in this cartoon, you can see this is the open state, basically. This is the microfluidic channel. This is the control channel. And when you close it, you can just cut the fluid flow inside of the channel. So, as a more detailed explanation, in our device first, we are trapping the cells inside of the proliferation chambers because we need to make single cell. We need to make analysis at the single cell level. Therefore, we need to capture single cells, and we have 8 parallel channels. It can be increased also, 16 to 32. But for the proof of concept, we just started with the basic version. And after trapping the cells, by using some bypass flows nearby the trapping chamber, we are just supplying a constant medium, because in cell culture, you should constantly supply fresh medium overnight or some 3 days, 4 days. It depends on how long you will perform the experiments. And after we observe division, by using T-junction structure, we are just separating the sister cells in this area. After separation, we have 2 options. First, we can relocate the sister cells in order to track the second generation, because in the first generation, single cell, double. If we want to track the second generation, we can just basically, by using this feedback loop, we can relocate the cells inside of the chambers and we can keep growing the cells. Or we can just extract the cells by pushing the sister cell towards the extraction area, extraction mats. So, this is the general setup of this device. We needed to have a microscope incubator because cells need certain environmental conditions to grow, like 37 degree, 5% CO2, and some certain level of humidity. For that purpose, we made a custom design microscope incubator. Inside of it, we have our microfluidic device enclosed by the CO2 chamber. And we have some control setup. In order to control the valves, we made a custom design PCB board, and it is connected to a custom design program in order to automatize the experimental process. And in here, for example, we have different subroutines. For example, cell capture, medium washing, or cell tracking. For example, in here, you can constantly get the cell image, and you can just report the division times for the statistical purposes. Or for some of the population of cells, division time is very important. You need to separate the cells just after division, for example. And this is the basic workflow of the device in terms of microfluidics. First, as you can see, we can track the single cells. And after a while, for example, for stem cells, this time is nearly 18 hours, 16 hours. For the other mammalian cells, it can goes up to 1 day, 24 hours. And after that, we are just separating the sister cells. And in the extraction area, we are just extracting the cells, because the one part of the this project is we need to understand the differences between the sister cells. For that purpose, we need to extract the cell, lyse it, and make some RNA sequencing analysis. So here you can see a cell cluster. First, we made an experiment with the model cell. As you can see, it keeps constantly growing. And after 2 days, in terms of area, we had a constant growing line. And we just optimized the certain medium circulation rates, and we can see more than 2 generations for a single cell. And for this cluster, for example, we start with the 5 cells, we can issue 24 cells after 48 hours. And as an advantage of microfluidics, we can constantly image the cells while they are growing. So after that, we just focused on our primary experiments, which are stem cell experiments. In here, we use two different types of stem cells. Basically, these are chicken progenitors, but one of them is immortalized cells, and the other one is the real chicken progenitors. And for the both of the cell type, we can achieve the proliferation rates around 90% and 80%. And as you can see, after around 18 hours, we can have divided cells here and for the other population also. So the other part of the project is relocation of the cells. In here, we observed one small problem because, for example, especially for this type of cells, they tend to stick together after division. So for that purpose, we applied an apoptose treatment. So this is also another advantage of microfluidics because you can apply some chemicals. In the future, for example, you can also change the surrounding medium in order to observe the effect of the medium on cell proliferation, for example, and it worked quite nicely. And as you can see, after around 1 hour, you can have the separated cells and you can relocate them to different chambers in order to track more than one generation. So as a final step, we extract the sister cells. In extraction part, it is hard to see from bright field image, so I would like to explain with the fluorescent image. You can see the single cell, and after extraction process, we can make sure that we just get the cells by analyzing the image. And the challenging part of this project is for RNA sequencing, we need a volume, a very small volume, extraction volume, which is half microliter, like 500 nanoliter, but we were able to achieve this volume because when we made the analysis, downstream analysis, we obtained the similar results with the FACS sorting. These are just control group sorted by FACS. And in terms of data quality, it is this project was a collaborative project with a French partner, so they did the analysis, but I can briefly explain about data quality is dependent on the number of molecules you can get after lysis. So most of them are near the quality of the FACS sorting, and you can see the clustering analysis. Most of them are related with the FACS sorted cells. So this was like a proof of concept study that we can grow single cells, separate them, extract from the device, and we can make some RNA sequencing analysis. And the quality of data is similar with the FACS sorting, which means it is not harmful for the cells, all the project. So from now on, I just want to talk about some flow cytometers, microfluidic flow cytometers. So first one is high-throughput and real-time flow cytometry for rare cell quantification. So I just want to talk about briefly what is flow cytometry. Flow cytometry is basically like a benchtop instrument. Here you can just, by using sheet fluid, you can just focusing the particles or cells that you want to analyze. And by using different laser wavelengths, you can get this forward scattered data or side scattered data. And by using some digital processing, most of the cases by using a computer, you can get some quantitative information about the cells or some molecules. So it is considered as the gold standard for analyzing the cells. So it has very sensitive detection because they are using state-of-art detectors like APDs, PMT arrays. And the throughput of the flow cytometry is also generally high. It is from 10,000 to 50,000 events per second. However, the disadvantage of flow cytometry is, although analytical throughput is very high, volumetric throughput is around 10 to 100 microliter per minute. So therefore, if you want to analyze some rare cells in a, for example, blood sample, it is not suitable because if you also consider the dilution factors, for example, you want to analyze 5 ml, 10 ml of blood, it will take more than 1 day because you need to dilute maybe even 100 times. And with this, by considering these flow rates, it will take more than 1 day. So therefore, it is not suitable for analyzing rare cells, for example, CPCs. I will talk about a little bit later, but the CPCs. And this another disadvantage is it is costly, bulky, and requires trained person to operate. And it is not easily achievable from the labs. So therefore, we designed. First, I want to talk about the viscoelastic focusing that we will use in our system, because I mentioned about the sheet fluid of the flow cytometer. So by using viscoelastic focusing method, it is a little bit complex phenomena, but basically by controlling the inertial, by controlling the Reynolds number and Weisenberg number, you can control the inertial and viscoelastic forces. And by using some viscoelastic polymers, you can achieve different kind of regimes and different kind of focusing planes. And with this technique eliminates using of the sheet fluid. So therefore, in microfluidic cytometers, recent years, this technique is being used commonly. For example, you have some cell sample. After a certain length of the channel, you can achieve plane focusing or single screen focusing. And this is very critical for analyzing because cells need to be in the same plane if you are analyzing, for example, if you want to make image analysis or fluorescent analysis. In here, for example, even subcellular features can be analyzed by using a microfluidic imaging flow cytometer. So therefore, in our system, we designed this kind of a setup, which includes a microfluidic device. And we coupled some simple optics. And in the microfluidic device, the key feature is we are just focusing the cells on the plane. And by using some basic objectives and filters and some cheap CMOS sensor, we can achieve the fluorescent intensity of the particles. And we were able to achieve a throughput of the 500,000 events per second. And more importantly, our volumetric throughput can go up to 6 ml per minute, which is much more than the flow cytometry when we consider it is just 0.1 ml per minute.
But not the laser? What type of laser?
This is just 488 nanometer laser. Yes.
And also about autofluorescence, is it better than the flow cytometry? How?
Yeah, we made a sensitivity analysis. I will talk about it in a minute. Yeah, I hope it will answer your question when I talk about it. So, and basically, this is our algorithm for detecting the fluorescent intensities. This is our background signal, but this is mainly caused by some laser reflection. Back reflection. It can be decreased, but you need to have more sophisticated design. And we are just modeling the background. And after using some signal processing, we were able to detect the peaks of the particles, and we can obtain the histograms. So, and by designing a custom written program, we can. Yeah, we can just obtain the histograms in real time. So in order to quantify the performance of our system, first we made experiments with the fluorescent beads. So we gradually increased the concentration of the beads from 1 million to the 4 million. So in here we observe that after 4 million beads per ml, we have some overlapping in the signal. Therefore, our throughput is limited nearly 500,000 beats per second. And another metric when we are considering flow cytometer is coefficient of variability of the intensity. And this is the flow cytometry level. It is around 22 for this sample. And we also have consistent results with the flow cytometry. So after that, we made cell experiments, which are our primary purpose. In the case of cells, in contrast with the beads, cells has much more smaller fluorescent intensity than the beads, because beads are very commercial and bright. But cells, we needed to optimize the exposure time. Exposure time is basically means how long your sensor is gathering the light. So, which means if you increase the exposure time, you are gathering more light. But as a trade-off, this will increase your motion blur. So then you need to decrease your flow rate or the concentration. Otherwise, you will have some overlaps. So in order to characterize this phenomena, we just use different exposure times. And we observed that in smaller exposure times, the concentration we made, this is around 1 million cells per ml sample, because we measured with the facts. And below around 100 microseconds, we couldn't measure the correct concentration. This is due to because, as I said, cells are less brighter than the beads. Another thing about the exposure time is if it is too long, you are getting less light, so you cannot detect very dim signals. Therefore, we decided that we also compare with this CV of intensity. As you can see, it keeps increasing until 200 microseconds. After that, it doesn't provide extra advantage. So when we consider these two graphs, we decided that 200 microsecond exposure time is the optimal for the cells. So, by the way, for the beads, for example, we were using 6 microseconds exposure time. Therefore, we need to decrease the flow rate, but still we could achieve a throughput of 75,000 cells per second in real time. And also our flow rate was the 3 ml per minute. And you can see the basic histogram, a simple histogram of the cells obtained with this device. So after that, we just made some sensitivity quantification. For that purpose, we use calibration beads, which are already have predefined intensity values. For example, this graph is gathered by the flow cytometry, and this is our setup results. So flow cytometry, when you look at, when you observe the data sheet, the sensitivity, the detection limit is around 100, 200 MESF units because of they are using state-of-art detection sensors. They are, but as a cost, they are very expensive. So, but when we consider our system, we just use 4 different intensity of the beads. The smallest one was about 3,000 MESF units. And when we decrease the intensity of beads, we see we are hardly detecting the beads, but still by using some signal processing, for example, this is the raw data from the number 1 bead population. We were able to detect the number 1 beads, which are 3,000 MESF units. This is higher than the facts, lower sensitivity than the facts, but when we consider the biological applications, most of the applications exhibit 30,000 MESF units or above. So it is enough for analyzing most of the biological samples. So after that, we just focus on our primary purpose, which is rare cell detection, where we can use the real advantage of our system due to the high flow rates. So in that purpose, we just spike fluorescent cell into the blood. These cells just fluorescent cell lines, and these cells we just derive from a real patient, brain tumor cells. And when we spiked into different ratio from 1 to 10,000 to the 1 to 1 million, even 1 to 1 million ratio, we were able to detect the cells, and we were able to detect the cells around 10 cells per ml in the blood. So I just want to move on this cell sorting system for the rare cell enrichment. Actually, this setup is like an improvement of the previous project that I have already talked about. So in our project, our motivation was maybe most of you have already know about, but CTCs are basically circulating tumor cells. And when you have primary tumor, if the cells are escaping from the primary tumor, it can go into the bloodstream, and it can cause some metastasis. And the challenge about the detecting CTCs, they are extremely rare. For example, 1 to 10 CTCs per 1 billion blood cells you can find. So there are some FDA-approved products or devices, for example, cell search system. But the disadvantage of this system, they have very low recovery rate or low purity rate. It is based on antibody-based system. So recently, some microfluidic approaches are developing. For example, this approach is like an ETH approach. Basically, you are just making tiny aliquots of the blood, and you are analyzing each aliquot. So this device achieve a higher purity, higher recovery rate. But the problem about this device is this is just closed device. So because after you get the detected CTC, you can make you want to make a downstream analysis by extracting the CTC cells. For this system, it is not possible. And another disadvantage is this system still has some complex setup, which are expensive and not easy to implement. So in our setup, we designed a microfluidic chip. It is basically a sorting chip, and we coupled with an optical system. And in this project, we just use an FPGA board. FPGA stands for field programmable gate arrays. These are very specific type of IC circuits, different than microcontrollers. You can program as an arrays, and you can make some programmable interconnections between the arrays. Therefore, you can do parallel programming very easily with this, because in our case, we need to have a parallel sorting mechanism. So it should do in real time with the circuit, and it should trigger the valves in real time. So basically, with our sensor, we are just detecting the fluorescent cells, and you can just apply some threshold level or some quantification. And after that, we are just sorting the intended cells. For example, this is our detection area, and this is our sorting region. In sorting region, basically, we are just triggering the valves that I have talked about it. These are the same collect valves mechanisms. So you can either collect the cells, or you can just, if you have blood cells, you can just open the waste area, and you can just collect the blood cells from the waste region. And in here, we also use the viscoelastic focusing method. So as you can see, these are the solution without PEO, without the polymer. And as you can see, they are just streaming in a random way. But if we use the polymers, PEO, viscoelastic focusing, it is also also tried with the different concentrations. In higher concentrations, even we have like a single stream focusing. This is very critical for sorting the cells because our valve mechanism, you need to have cells one by one. So, and in order to quantify our system, first we made experiment with the fluorescent beads. We are just mixing fluorescent beads with the non-fluorescent ones at different ratios. And the concentrations vary between 10 to 100 beads per ml. And we can achieve an enrichment factor of 2000. And also, our flow rate was higher than the fax. It is like a half milliliter per minute. It is like 10 times higher than the commercial fax. And when we consider the recovery rate, for all the cases, we have recovery rates more than 90%. So after that, we just focused on our primary purpose, which is CTC, circulating tumor cell sorting. So for that purpose, we just typed the brain tumor cells gathered from the real patient into the blood. And in this case, we just use the realistic concentration, which is 1 to 10 to the 8. And the advantage of our system is our volumetric throughput of our system is high, so it will take around 10 minutes to run the 5 ml of blood sample. Yeah, if you want to dilute, the dilution factor about 5 times. So if you want to analyze 1 ml of blood, let's say, it will take 10 minutes. So, for example, this is therefore you can just sort the same sample more than one time, like a cycling. I mean, for example, we are just putting our blood sample from here. We just collect the sorted sample and put the sorted sample again from the inlet. So with this mechanism, for example, this is the initial sample. This is after first round of sorting. After 3 rounds of sorting, which is basically 30 minutes, we can achieve 72 million times enrichment, which means 72% of the population is now CTCs. This is like a huge number of enrichment when we compare it to literature also. And the other thing is about cell viability, because as I said, you need to do downstream analysis as an advantage of our system. So cell viability is very important after sorting, because maybe you need to do some subculturing of the sorted cells. So for that purpose, we just use a control sample and first round of sorting. When we compare the viability of these two cells, they are still high after sorting even. This is 95% of viability we achieve, which means the sorting mechanism is not harmful for the cells. So as a last project, I want to talk about the smartphone-based imaging flow cytometry for high-throughput single-cell analysis. So I just want to talk about imaging flow cytometry really quickly. So imaging flow cytometry is very similar to conventional flow cytometry. However, you have some imaging modalities in imaging flow cytometry. So therefore, you can gather some spatial information about cells. So what is the advantage of this? For example, you can acquire the images of the cells. You can use up to, for example, 12 channels in this example, for example. You can have bright field image, some fluorescent image, and you can extract many features from this image. For example, area, intensity, granularity, and other parameters. And you can map these features, and for example, you can make some analysis on the bright field images as well. By using some, for example, deep learning methods, you can use it as a diagnostic purposes, for example. But the disadvantage of flow cytometry, imaging flow cytometry is their throughput is limited to 2,000 to 5,000 cells per second when we compare it to conventional flow cytometry. Because you are basically gathering image data, it is like a two-dimensional data, and in terms of memory considerations and image processing consideration, you have less analytical throughput than the conventional flow cytometry, and also it doesn't overcome the disadvantage of flow cytometry in terms of cost and bulkiness. For that purpose, in this project, we designed a smartphone-based cytometry setup. Basically, we coupled smartphone with some simple optical setup, and we developed a custom software so it can acquire the image of the particles of the cells, and it can process in real time, and it can classify them. So you can see the basic image pipeline of the image processing operations. First, we are recording the cells, and we are just using some morphological operations. We can just detect the cells. So in order to quantify our system, first we use the beads, polystyrene beads. So this is the raw image, and this is after image processing. We use two different mixture of beads. In this mixture, we have 10 and 15 micrometer beads, and in this mixture, we have 10 and 12 micrometer beads. So in both cases, we have consistent results with the flow cytometry, and therefore, in this case, for example, we were able to distinguish the beads which have a size difference of 2 micrometer, and our throughput is also 50,000 beads per second. So this is like a basic histogram of the beads. I mean the number of beads, but the most critical advantage of imaging flow cytometry that we want to use the spatial information of the cells. So because in terms of sizing, you can do it with the flow cytometry even higher with higher throughput. Therefore, we just developed a machine learning algorithm that run on the smartphone, and this is the basic pipeline of the algorithm. First, we are recording the cells. In this case, we just use UCAT and EL4 cells. I'm not sure you can see, but these are similar size cells, but EL4 cells have more granularity than the UCAT cells. I mean, the subcellular features are different. So after image recording, we are just detecting the image, individual cell image, and after that, by using the pre-trained model, we are just retraining our machine learning algorithm, and we just issue the classification results. So as an experiment, we just did 3 different experiments. First, we just use the single population of cells, UCAT cells and EL4 cells. This is the sizing histogram of these cells. As you can see, they are highly overlapping in size, especially in this cell mixture histogram. You can see it is impossible to distinguish these 2 kinds of cells by using a size-based approach. And this is the output of our machine learning algorithm. So we achieved the classification rates more than 90% for the individual cells. And this is the known population mixture. And when we applied our algorithm to the cell mixture, we were able to classify these 2 different types of cells. So as a conclusion, we made, first I just introduced some cell manipulation methods. So I just talked about label-free cell manipulation and single cell trapping and proliferation experiments. And we were able to manipulate single cells very efficiently, both in label-free way and using some mechanical methods. And after that, we just implemented some high-throughput cytometries. So it can achieve a throughput, analytical throughput of 500,000 beats per second. And it can go up to 6 ml per minute flow rates. And we can successfully analyze the CTCs or sort and enrich the CTCs in the blood samples. And lastly, we just use an imaging flow cytometry approach in order to classify machine learning, in order to classify the cells have similar size but different morphologies. So as a feature work, for example, the sensitivity of cytometries can be increased more by using more sensitive sensors and by improving the optical setup. And we can add, for example, more parameters into our cytometers. For example, this is the deformability cytometry. We have some preliminary results. This is basically you are deforming the cells and you are imaging the cells, deformed cells, and you are just extracting the deformation metrics. Because deformability is another parameter for the cells, distinguished parameter. And we can also make a machine learning-based cell sorting by improving our machine learning algorithm. We can design a cell sorting platform. So I want to thank you for your listening. If you have any questions, I will be glad to answer.
Thank you very much. Any questions from the audience? And we can try. I guess we won't be able to hear the Zoom people, or they should type something over.
Maybe we can from the.
Maybe. If they have, we can give it a try also.
Yeah, they can write from the chat.
Or
Yes,
we
we are.
can actually hear the questions.
Okay.
If they just unmute themselves.
So if they present, let me know.
Yeah.
Okay, any questions? So these 5 studies are essentially all of them was your PhD work?
Most of them, 4 of them. One of them, the first one is like a last stage of the master's and master's studies. Yeah.
Oh, but I wasn't sure. So in ATH, you just put like 5 different publications or 5 different chapters, and
Yeah,
then make it a book, and that's
Make
a
it
PhD.
easy, yeah.
That's a PhD.
Yeah.
Okay.
this is how it works. Yeah.
All right. Go ahead.
I want to ask a question about multi-layered microfluidic device.
Yes.
You say that you use PDMS and quake valves for backflow, preventive backflow, but we know that PDMS for the PDMS production we need clean room and it's a long process. And sometimes it's a problem for rapid production of microfluidics. How we can handle this problem? What do you think?
Yeah, good question. Actually, in PDMS, there are 2 fabrication processes. First, you need to fabricate mold for the PDMS, and after that, it is very straightforward process. You are just, I can give an example of baking the cake because you are just mixing the curing agent and PDMS. So, fabricating the mold in our cases, it is like a 1 day, half-day process, and once you fabricated the mold, you can use the same mold like 10 times, 20 times, and each mold you can fit, for example, 10 devices, 20 devices. So in each fabrication, basically you will have 10 devices, 20 devices easily. For the mass production, maybe yes, you need to investigate different techniques, but for experimental purposes, it is very straightforward.
Thank you.
So I'm not sure. Do we need an absolute clean room for all applications?
For the mold fabrication, yes, but not very advanced clean room.
Right.
We have a very small.
People do it effectively in a relatively, I guess, film boot or biosafety chamber or something.
Yeah.
I've seen it. Because we were always wanted to utilize all clean room for that purpose.
I saw it actually. Yeah, this
But-
clean room is
but-
very nice.
They said we don't need it. They do it within our lab, so we don't.
Do you
have
need nano-imprint lithography, or is it
More raise your voice.
lithography, nano-imprint lithography system, or just
Actually, in our cases, our minimum feature size is around 10 micrometers.
so you don't?
So we didn't need, we just have some simple masks, not the chrome masks. But we had also like a nanoscribe device for some nano fabrication process, but all of the projects are just basic fabrication methods.
Go ahead, Rigo.
I was wondering whether you utilize a technique that's widely used in searching the stem cell populations. It's called site population.
What do you mean by that?
So the site population technique is you use a UV laser and try to find out stem cell population using some type of a dye called Hertz. Just wondered whether you tried the RTCS system or has it been?
Oh, okay.
Utilized.
Yeah, it can be, but mainly we just focus on CTC detection in our cases. As a feature, we were just planning to focus on some exosomes or extracellular vesicles as a nanoscale, but not the stem cell study. But yeah, you can do that.
Is it something that we can purchase or is we have to collaborate?
No, sorting is, we are planning to file a patent. So, but commercialization is very long process. So for now, I can simply say no, but hopefully in the future, in near future, yeah.
So ETH has active pathways for commercialization and access.
It's like that, especially for the sorting system. Now we are collaborating again, like maybe the doctoral studies, we are just finding a funding application.
Okay.
So this is our purpose, like in 1 year, 2 years, we want to make it commercialized. Yeah.
Okay. Go ahead.
How much automation is currently in the processes like separations and stuff? Do you have to go in?
For now, yes, you need to interact still, but it can be fully automatized. Yeah, because the first part of the project is the automatization of the project, but to be honest, the other cell proliferation part took a very long time. Right. So we just focused on this part more, but it can be developed, yes, fully automatized experimental process, yes.
Go ahead.
Thank you very much for the nice talk. So especially your talk about flow cytometer is very interesting, I would say. If you produce it in a cheaper way, I would buy one. We need it, I think, at the institute as well.
Everybody asking for flow cytometer.
Yes, that's very
Not
important. That's not cheap, I would say. That would be very promising. What I would like to, you're using PDMS chips, right? For all of them. And micron size, as I see. Did you ever use this for droplets formation?
Yes,
Or
actually,
emulsion?
yeah, our group's main expertise is droplet microfluidics actually, but I am not included in this kind of projects, but my lab mates were using it, especially for enzymatic reactions or encapsulating cells or even sorting the cells. First, they encapsulate the cells into droplets and using some electrical methods, for example. Yeah, they were.
Okay.
So
That's what I'm interested in. That's why I'm asking this. But are you also in the group using chips other than PDMS? Because I know this is a very hot topic. PDMS is great,
yes,
but it has also some, from the material perspective, now I know some,
biofouling, for example.
yeah, probably.
Yes.
Transparency.
That could be also, or solubility actually. So if you want to make, for example, emulsions in there, so you can also dissolve the PDMS at some certain point. Are you also, I know the
Yes,
deMello is big. Are
Yeah,
you
one
this direction as well?
of my colleague.
Green deal folklore.
You can get some trick.
Yeah, one of my colleagues was producing Teflon chips.
Okay, yeah.
In order to overcome this biofouling, yeah, in our group, yes. But mainly PDMS or some paper microfluidics, but small amount of paper microfluidics.
Paper microfluidics will be even cheaper.
Yes,
That was
basically, Optimers and others.
Yeah. See, the cheaper it is, the more motivated I am.
Yeah, I can understand.
Exactly. So,
Okay.
thanks.
Well, any other questions? We can, if there are more questions, we can close the formal part on time, and then ask maybe more intimate and fun questions after the formal part of the seminar is finished. So we encourage the face-to-face participation in seminars. More exciting and social interactions are gonna come next after the seminar. Everybody. Look, I'm barbecue, happy hour. So we officially close it for the online viewers and start the more fun part afterwards. So next time,